that is told in the class. Okay, so let's start. One second, just give me a second. Yeah, so let's start today's session. Photosynthesis in higher plants. So we know that photosynthesis, photo means light, synthesis means production. So using light, all the autotrophic plants, especially photosynthetically autotroph. So these particular plants or organisms, they know to convert this light energy into chemical energy. So which today we know the proper representation of photosynthesis, where we write for photosynthesis we need carbon dioxide and water, and then eventually it gives rise to formation of glucose, and oxygen is evolved as a byproduct. Now, to understand photosynthesis, it was a long process and it has to be done to meticulous experimentation over the years. So it was not one step process. So let's understand the early experiments. In our textbook, in our later classes, in our like fifth or sixth or seventh standard, we would have heard about this variegated leaf experiment. Maybe the name confuses you, but the concept is very simple. We take the leaf, and then one of the leaf, we cover it with black paper. Partially, half of the leaf is covered with black paper, and we expose it to the light. So now, whichever the part that is exposed to the light, it will show the presence of starch, and whichever that is not exposed to the light, that will not show any presence of starch. So we understood that whenever the light exposed region of the plant is producing starch when it is given light, correct? So this was variegated leaf experiment. Other one was half leaf experiment. So whenever you hear about half leaf experiment, remember that you talk about KOH or potassium hydroxide. The thing about potassium hydroxide is whenever it is just dipped in a cotton ball and then put in a test tube, it absorbs all the carbon dioxide. So now let's say that one half of the leaf is outside the test tube, other half of the leaf is inside the test tube, and there is a KOH soaked cotton that is present within the tube. Now what happens is KOH absorbs all the carbon dioxide. Now there is no carbon dioxide and there is no photosynthesis happening, or the production of food is happening. And the rest of the portion which is exposed to the normal carbon dioxide, they start producing starch. So this one tests positive for starch, and the one that is inside will test negative. So this is of carbon dioxide. Next one is Joseph Parsley. A very important and significant discoveries have been made by Joseph Parsley. One second, yeah. Parsley in 17 what he did was he put a candle in a jar, and he observed that the candle got extinguished, or it got off after a certain period of time. The same fate where it died. Now he understood that when he put the mice and the candle together within a jar, somehow they both died faster or extinguished faster. He somehow concluded that something that is the plant within this jar, and he observes that all of them were living for a longer period of time without dying. So then that led to the significance of oxygen and carbon dioxide with respect to Parsley's experiment. Now after Parsley, hold on, yeah, after Parsley, any photosynthesis, and they did not purify the air, whereas the ones that are in the sunlight, okay, so they start purifying the air, or they started producing the oxygen. And he also continued his experiments where he took aquatic plants and he showed that whenever there is bright light, they start forming small bubbles, and these bubbles were nothing but oxygen. Okay, so he is the one who identified oxygen. Next one, Julius van Schaas, what he proved is the resultant of this photosynthesis is glucose. First glucose is produced and then it is converted into starch or stored as starch. And he also identified there are some of the chlorophylls that are present as special bodies, and within the chlorophyll there are chloroplast which act as photosynthetic pigments. Okay, now coming to experiments conducted by Eagleman. So all these experiments are very important, students, so please read it once and write it down. Now Coming to Eagleman started splitting the lights, okay, into spectral components. So we know that we have whip jar when the white light is passed through the prism. Now what happens is when it is passed through the prism, now the light is split into different lights like blue light, red light, etc. Now he uses green algae such as cladophora. So he places in the suspension of an aerobic bacteria which uses the oxygen. Cladophora, when it photosynthesizes now, this bacteria goes and grows near where the oxygen is evolved. And this particular green algae was evolving more and more oxygen, especially at the blue and red light area. So this blue and red light regions is where more photosynthesis was happening. So this region was called as action spectrum, where there is a lot of photosynthesis is happening. Okay. Now, coming to the some of the experiments conducted by Cornelis Van Niel. So Van Niel, what he does is he conducts these experiments on some of the bacteria such as purple and green bacteria. Is very significant for what he did later. He demonstrated that photosynthesis is a light-dependent reaction, and where hydrogen is very important for it is an oxidizable compound. You can oxidize hydrogen. So it also reduces carbon dioxide to carbohydrate. That's what he said. And he also gave an expression where anything that can be with association with hydrogen, like H2O or H2S. Now here, this hydrogen and the O or S bonds are broken, and this is where it gets oxidized. And here, this is used for with the carbon dioxide and presence of light, we have the production of glucose and the evolution of oxygen or sulfur and then water. So his equation proved that we understood that not all organisms use carbon dioxide, water, and sunlight for the production of energy. There are certain purple and green sulfur bacteria which use H2S, where hydrogen is used as a donor. Here they don't produce oxygen, instead they produce sulfur or sulfate, etc. Okay. Now, once you have this oxygen that is evolved from the green plants, basically it is coming from the water and not from the carbon dioxide. That's what was proved by this Van Niel's experiment. That is why it's very significant. Okay. So the correct equation can be derived for the photosynthesis, where to complete photosynthesis, we need 6 molecules of carbon dioxide, 12 molecules of water. In the presence of sunlight, it gives us glucose, water, and oxygen as the byproduct. Okay. Now, coming to the chloroplasts. Now, we know that the site for the photosynthesis is chloroplasts, and within the chloroplasts, we have photosynthetic pigments called chlorophyll, which which participate in photosynthesis. Now, if you understand the structure of chloroplast, we know that it is double membrane bond. And within this inner membrane, you have a uniform matrix called as stroma. Stroma contains a lot of enzymes. You can see here, right? So the stroma contains a lot of enzymes which converts this glucose into sugar, or your or carbon dioxide into glucose or sugar. So here, since they participate in manufacturing this glucose, and it happens with the dependency of light, not directly on light. So that is why they are called as dark reactions. Whereas if you see within the stroma, we have thylakoids, and within this thylakoids, we have quantosomes, or we have chlorophyll pigments, where it knows to trap the light energy, and it synthesizes two important byproducts such as ATP and NADPH reactions. Okay, give me a second. Yeah, ATP and NADPH reactions. Sorry, byproducts. One second. So ATP and NADPH is produced with the resultant of light reactions, and in the stroma they have enzymes where they manufacture this sugar, and this is called as dark reaction. Okay, so now coming to the pigments of the photosynthesis. Okay, so one second. Yeah, yeah. Coming to the pigments of the photosynthesis. Now, what happens is there are several pigments that are present within the thylakoids, membranes of the thylakoids, such as chlorophyll, chlorophyll A, and chlorophyll B. And then we have carotenoids, we have cytochromes, cytochrome A, cytochrome B, cytochrome B6, and then we have quinones, and then ATP synthetase, which help in manufacturing of light reaction products. Now, when we talk about arrangement of these pigments, all the accessory pigments such as chlorophyll B, and then we have xanthophylls, and then we have our carotenoid pigments. They are called as accessory pigments, and they are arranged in hundreds of number, where they harvest a different wavelength of light and they act as antenna. The role of this accessory pigments is to absorb broader range of wavelength of light and also provide protection to your chlorophyll A, because if the large amount of light is incident on your chlorophyll A, now the chlorophyll A disintegrates through uh, photo reaction. It goes into a photo reaction where it gets degenerated. Now, coming to the arrangement of your pigments, I told you it forms this antenna, including all the pigments except for chlorophyll A. Now, the chlorophyll A alone forms the reaction center. Based on the type of chlorophyll A, we have two types of photosystems. One is photosystem 1, and then we have photosystem 2. Now, the difference between photosystem 1 and the photosystem 2 is the type of wavelength they are having the capacity to harvest. So, if you talk about photosystem, you can see here, right? So, coming to photosystem, hold on. Yeah, here it is. Yeah, you can see here. We have one photosystem that can harvest around 700 nanometer of wavelength of light, and another photosystem that can harvest around 680 nanometers wavelength of light. Now, when we talk about photosystem 1, now photosystem 1 contains the pigment which can harvest around P700, that is 700 nanometer of length of light. And photosystem 2, on the other hand, it can harvest up to 680 nanometer wavelength of light. This is where they can perform optimum or highest. And along with that, they have the accessory pigments, which forms the reaction center. Now we have your photosystem 1 and photosystem 2. They undergo a specific reaction, which we call it as cyclic photophosphorylation and non-cyclic photophosphorylation, where they produce NADPH and ATP as the byproducts, which are very, very important for further reactions of photosynthesis. Okay. Now let us talk about the light reaction or the uphill reaction. Okay. Now to understand the uphill reaction, we need a Z scheme. See, we call this a Z scheme because we have two photosystems that are placed. One is the photosystem 2, and then we have the photosystem 1. Now, in photosystem 2, what happens is first the light is getting incident on the photosystem 1 and photosystem 2. Now, what happens is the electrons that are present in the photosystem 2, they start getting excited and they start jumping into the higher orbitals. Once they go into the higher orbitals, these electrons are picked up by several electron transporters, okay, such as yeah, we have several excuse me, yeah, we have several what do you call electron acceptors. So these electron acceptors, they carry these excited electrons. For example, yeah, they carry this electron down the gradient. So they go down the redox potential, and they use this electron transporters to jump from one electron transporter to another electron transporter based on the redox potential, and then finally they give up their electrons to photosystem 1. Once the electrons from the photosystem 1 reaches the photosystem 2 to photosystem 1, now with the incident of 700 nanometer of light, again the electrons get excited. Here they start moving up again the redox potential. Eventually what happens is these electron transporters, they reduce this NADP plus to NADPH. So that is when you have your NADP H. Okay, now you can see here. So this thing happens in a non-cyclic pattern. That's why we call it as non-cyclic photophosphorylation, or it is also called as Hill and Bandel reaction. Okay. Now here what happens? First it goes from photosystem 2, and then it gains the energy, and it is picked up by some of the electron transporters called as pheophytin. Okay. So these are electron acceptors. Okay. And then for example, plastoquinine is another example for electron transporter. So they pick it up and they pass it down to cytochrome F, and then cytochrome F to the plastocyanin, and plastocyanin it handovers to P700. Okay. So when it comes to P700, again it gets excited and the electrons pass to FES, FD, and then finally it is NADP plus is reduced to NADPH. So the byproduct of non-cyclic photophosphorylation is production of NADPH. Now coming to cyclic photophosphorylation, where does it happens? Now if you look at the thylakoid, okay, we have the membranes of the thylakoid, and then we have an extension of the thylakoid which you call it as stroma lamella. So in stroma lamella, what happens is we have only one photosystem, that is photosystem 1. There is no photosystem 2. Now what happens is the movement of the electrons, like how it has happened from photosystem 2 to photosystem 1 in non-cyclic photophosphorylation, this doesn't happen in for cyclic photophosphorylation. In cyclic photophosphorylation, what happens? Here, it returns back to the same photosystem again and again. So the question is, where does it happen? It happens in the stroma lamellae. How many photosystems it has? It has one photosystem, which we call it as photosystem 1. How does it happen? The light incidence on the photosystem 1, the electrons get excited. After it gains the excitation, so these electrons are transported through our electron acceptors such as FES, and then it is going to the ferredoxin, which is another electron acceptor, and then cytochrome B6, and then cytochrome F, and then from cytochrome F, it again moves down to the plastocyanin. Okay, again moves down to the plastocyanin, where plastocyanin gives back to P700 again. So this is called as cyclic photophosphorylation. Now this, why are we discussing this? The reason why we are discussing this, the light reaction happens in the chlorophyll. Correct, right? The light reaction is happening in the chloroplast, and in chloroplast especially we have thylakoids and we have pigments arranged. Here they are participating in a reaction called as phosphorylation. So phosphorylation is the way where the energy that is released is stored in the form of chemical bonds, especially in the form of ATP. Here we discuss two types of phosphorylation reaction. One is non-cyclic photophosphorylation reaction. Other one is called as cyclic photophosphorylation reaction. Here in non-cyclic photophosphorylation reaction, we understood that there is production of NADPH, whereas in cyclic photophosphorylation, for some reason, there is movement of electrons from photosystem 1 to back to photosystem 1 again, and this is happening in the stroma lamella. Okay, right? So we understood this photophosphorylation. Now, when it comes to the phases of photosynthesis, photosynthesis is a photochemical and a biochemical reaction. So, photochemical phase is the light reaction or the Hill reaction, whereas biochemical phase is the dark reaction, or is also called as Blackman's reaction. So, as we have discussed, the names for this reaction is given based on how the first reaction takes place. In light reaction, what is happening? We understood that somehow these electrons are getting excited and they're transported. It leads to the formation of ATP and NADPH. Now, this ATP and NADPH that are produced, they are very, very important for the dark reactions to take place. So, this dark reaction is where ATP and NADPH are used for the synthesis of glucose. Okay. Now, coming to what is happening importantly in your, light reaction. So, one of the key steps that happens in the light reaction is the presence of light. So here what happens is two things are produced. One is ATP and NADPH. We talk about ATP and NADPH, how they are produced in a certain while. Now, how do they eventually end up in producing NADPH? So now the light incidence on the photosystem 2, we told that it gets excited and reaches to higher states of photosystem 2. It goes through series of electron carriers such as pheophysin, right? And then plastoquinone, and finally it comes to the cytochrome B, and then it goes to the plastocyanin, and plastocyanin to P700. Here P700, again it goes, we understood this. How did this happen? Why did we require light? Light is used for breaking the water molecules so that water is split into hydrogen, oxygen, and electrons. So water becomes the very important source for photosystem 2 to supply its electrons. Okay. So the water is used for the breaking down. So water is broken down into hydrogen, oxygen, and electrons. So these electrons, such as chlorine, calcium, and magnesium, is required during the process for the transportation of these electrons down the redox potential. Understood? So as these are, as the photolysis contributes to the supply of electrons, now that is moved down the order, it reaches the photosystem 1, and then it gets excited. Eventually what happens? The ferredoxin reduces your NADP plus to NADPH. Now remember students, this particular reaction is very, very crucial for us. Why? Because it produces one of the key byproducts of the light reaction, that is the formation of NADPH. Okay, next one. Now coming to how the assimilatory, that for example I told you that the photolysis of water gives you the electrons. Okay, but if you look at the photophosphorylation, the way in which the electrons are picked up and trapped within the P700, so these electrons are expelled by the P700 itself, and again it is picked up by the FES when it comes to the non-cyclic photophosphorylation, right? So one process helps in production of NADPH, other process helps in the production of uh, you know ATP. Okay, now another important thing that we have to understand is, we understood that NADPH production takes place. Now I'm mentioning about the production of ATP. Now how does the ATP production takes place as well? So this is our question mark. Now, to understand the production of ATP, we have to understand there are two important organelles that participate in production of ATP. One is our mitochondria and the other one is our chloroplast. How do mitochondria produce ATP? Mitochondria is called as a powerhouse of cell. It has two membrane, outer membrane and the inner membrane. The inner membrane, it forms cristae. So within this inner membrane and the membrane space that is present between the two membrane, which is called as perimitochondrial space, here what happens, the proton gradient, okay, or the positive charge starts accumulating between this membrane space and then the space within the cristae. Now here, because of the proton gradient or the difference in concentration of protons, now the protons are pumped from within the membrane to the inside of the mitochondria. So this proton gradient is developed and this is used for the synthesis of ATP. Similarly, the same thing happens in thylakoids also. That is when the membrane of the thylakoid becomes a very, very important part when we discuss chemiosmotic hypothesis. If we look at the chemiosmotic hypothesis, we have to understand that thylakoid is a disc-like structure. It has a membrane, and within the thylakoid, we have the lumen of. Oh, sorry, we have the lumen of the thylakoid. Okay, this is the lumen of the thylakoid. This is the membrane of the thylakoid. This is the stroma. Okay, that is the outside of the thylakoid is stroma, right? Now we will understand the relationship of the H plus concentration in two regions. One is the lumen, other one is the stroma. Stroma is outside. I'll write it here. Stroma is outside the thylakoid and lumen is within the thylakoid. Now, there should be the establishment of hydrogen ion concentration. How does it happen? It happens in three important regions. It is very, very simple to understand. Look at this. We have the membrane of the thylakoid. Within the membrane of the thylakoid, we have photosystem 2. Now, we know that photosystem 2 undergoes photolysis. That is, it breaks down water. Now, where does the water breakdown takes place? It takes place within the lumen of the thylakoid. Within the inside of the thylakoid, what is happening? The water is broken down into hydrogen, oxygen, and the electrons. Now, the hydrogen concentration within the lumen increases, correct, in one place. Now, these excited electrons, this is the first stage. How does the hydrogen ion increases within the lumen? Now, we have water molecules. Water molecules are broken down within the lumen by the photosystem 2. When it's broken down, it releases hydrogen and it releases oxygen. Sorry, it releases hydrogen and it releases oxygen and electrons. When it releases hydrogen, the hydrogen is released is within the lumen of the thylakoid. Understood? Now, the second stage. Now, for the transportation of electrons, excited electrons from the photosystem 2 all the way till the electron transporters. Now, these electron transporters which accept the electrons, they are able to transport this electron through the membrane by doing something. What do? They take the hydrogen that is present in the stroma and they start pumping towards the inside. With the exchange of one proton, they are able to exchange the electrons. So the electrons are exchanged because of the exchange of protons. So when the protons are pumped from the outside, that is the stroma, into the lumen, now again the hydrogen ion concentration increases. Got it, people? Next one, the third case. In the stroma, what we see is there is a present of NADP plus and H plus. So because of this, they have a special type of an enzyme called as NADP reductase. Okay, I will write it. I'll write it here. NADP NADP reductase. Now, NADP reductase, which is present in the stroma, what it will do is it will take the hydrogen that is present in the stroma and it starts reducing NADP plus to NADPH. When you add hydrogen ions from this to this, what happens? It reduces the amount of hydrogen ion concentration in the stroma. What happens? Decreases. Because of 1, 2, 3 factors, the hydrogen concentration within the thylakoid increases to very, very large extent. Correct, no students? So when it increases to very, very large extent, now we know that we have created a proton gradient. Now, why do we call it as a proton gradient? In the stroma, the proton concentration is very, very less. But within the lumen of the thylakoid, the hydrogen concentration has become very, very high. Now, this gradient is utilized in the next process. Let's see what is that. Now, if you look at one second, if you look at the one portion of the thylakoid membrane, they have a special studding. Studding means they have something called as CF particles. CF particles, they are coming in two subunits. We call it as CF0 and CF1. Now there are like two components. Okay. Now they form the transmembrane proteins. I'll tell you what is transmembrane protein. The F naught or the CF naught, they are embedded. They form like a channel between the lumen and the stroma, like a pipe or the channel. So that is called as CF naught. And the CF1, it contains a special portion where the small changes or the structural configuration that happens in the CF1 activates this enzyme called as ATP synthetase. Now what is happening? The CF0, what we have here, it can detect high amount of hydrogen concentration in the lumen. Once it detects high hydrogen concentration in the lumen, it starts pumping the hydrogen, okay, from the inside of the thylakoid to the outside of the thylakoid. So once it starts pushing from inside of the thylakoid to the outside of the thylakoid, now the hydrogen ions have to pass through this another particle called as CF1. CF1 is to the outside of the thylakoid membrane. Now once the hydrogen ions cross through the CF1, what is happening here? There is a structural configuration in the ATP synthetase. Now this leads to the formation of ADP into ATP. Now, what does this do? ATP synthetase. For ADP, it starts adding one phosphate molecule and eventually it gives rise to ATP. Now you should understand how the ATP production takes place within our thylakoids. Correct, no students? How the ATP production takes place within our thylakoids. So ATP is done and NADPH is also done. Now, one second. Give me a second, students. All right. So today we have discussed some very, very important topics. And now we have to go for the next one, that is the dark reactions. Before we go into the dark reactions, we'll just look into some of the important things. Okay. Chalo, I think you should have understood by photophosphorylation or non-photophosphoryl, non-cyclic and non-cyclic and non-cyclic photophosphorylation. We produce ATP and NADPH, correct, right? So now we have the next step of the reaction, which is called as the dark reaction. Now, what happens in dark reaction? That is another important topic we have to discuss now. Okay. Now, I told you that the dark reactions depend on the products of light reaction, that is ATP and NADPH are very, very important. Now, this ATP and NADPH are used for a particular reason, and we will understand how. Now, whatever the enzymes that are required to conduct the dark reactions, they are present in the stroma. So there are two ways in which this dark reaction happens, which were represented by C3 pathway and C4 pathway, or we call it as C3 cycle and C4 cycle. Excuse me, yeah, C3 cycle and C4 cycle. Now we will understand one by one. What is this C3 cycle, or what is it called, C3 or Kelvin cycle? Kelvin is the one who discovered this pathway, and this pathway talks about how carbon dioxide is used for the manufacture of glucose. Now, they were trying to understand how this carbon dioxide molecule gets fixed. Understood? While discovering that, what he did was he started working on, okay, some of the important experiment where he started labeling the carbon dioxide with radioactivity. The radioactive, okay, C14 he used. When he used it, he understood that the carbon dioxide was first fixed as a three carbon molecule compound. See, you can see here 3PJ, which is called as phosphoglyceric acid. Phosphoglyceric acid contains three carbon atoms in the structure. Since it contains three carbon atom in its structure, that is why we call it as C3 cycle. Understand this. Carbon dioxide is fixed as a three carbon containing compound. That is why we call it as C3 cycle. Now, understanding this, which is the phosphoglyceric acid, which is a third compound. Now, they wanted to understand that, okay, if carbon dioxide is fixed as a phosphoglyceric acid, okay, I'll write it as 3 PGA, which is three carbon containing. Here, carbon dioxide is one carbon. Here, it is three carbon. Obviously, what you will think, 1 plus 2 is equals to 3, correct, right? 1 plus 2 is equals to 3. That's what you think. That's what even the scientists think. What did they think was, if carbon dioxide is fixed as a three carbon atom compound, then definitely there is a two carbon atom. See, 1 plus 2 has to happen. So they were finding which is this second two carbon atom containing compound that is helping our one carbon dioxide to become a three carbon containing PGA. But unfortunately, they did not discover any two carbon containing compound. They came across some strange compound called as RUBP. So RUBP stands for ribulose bisphosphate. So this ribulose 1,5 bisphosphate or ribulose 1,5 diphosphate, which we call it as. So this is a 5 carbon containing, okay, ketose sugar. It is a ketose sugar which is 5 carbon containing. So this 5 carbon containing sugar was helping our carbon dioxide to fix as 3 PGA, okay, or phosphoglyceric acid. Now, when you talk about Kelvin cycle, for each cycle, it can only fix one carbon dioxide per time. That means 6 times the cycle has to take place. Now, let us understand how this hexose sugar or the 6 carbon atom sugar is molecule is produced by this, okay? First, when we have carbon dioxide is fixed as a 3 carbon atom containing molecule called as phosphoglyceric acid. And for this, you have a very important enzyme called as rubisco, ribulose bisphosphate carboxylase oxygenase. See this enzyme? So this enzyme helps in the formation of phosphoglyceric acid. And this enzyme is abundant in nature. They're very, very high in nature. Understood? So now what it does is it converts this carbon dioxide into 3 carbon containing phosphoglyceric acid. Now here this phosphoglyceric acid, what it will do is, here the ATP is reduced to ADP, and in the same phase NADPH is reduced to NADP plus. Now you can observe that ATP as well as NADPH is used in the C3 cycle. So this 3-PGA is eventually converted into glycerol 3-phosphate. So this glycerol 3-phosphate, which is one of the molecule, is used for the formation of glucose, whereas one of the glycerol 3-phosphate, which is 5 molecules of glycerol 3-phosphate, is used for the regeneration of RUBP. So RUBP is always required for fixing this carbon dioxide. You see, RUBP, carbon dioxide combines 3-PGA. Where do we get this RUBP? RUBP we get through the 5 molecules of glycerol 3-phosphate, and the glycerol 3-phosphate undergoes regeneration process where this glycerol 3-phosphate becomes RUBP. So how does it happen? Again, 3 molecules of ATP is used and it is converted into ADP. This energy is used for the recovery of RUBP or ribulose bisphosphate. Understood? So what is Kelvin cycle? It is a C3 cycle. And what is the important enzyme? RUBP. What is a 5 carbon sugar? That is keto sugar, RUBP. What is the enzyme that fixes the carbon dioxide? Ribulose bisphosphate. Ribulose bisphosphate, carboxylase and oxygenase. And what does it fix into? Into a 3 carbon containing compound called as phosphoglyceric acid. Now what happens to this phosphoglyceric acid? Here the ATP and NADPH gets reduced into ADP and NADP plus, and this is used for the conversion of 3-PGA into glycerol 3-phosphate. One of the glycerol 3-phosphate is added for the manufacturing of glucose. Another glycerol 3 and the rest of the glycerol 3-phosphate is recycled back for production of or recycling of RUBP. Okay, again here the ATP is utilized. That is why light reaction are very, very important, people, for this process. Now, coming to, hold on 1 second, students. Yeah. Yeah. So now we understood that the C3 plants, there are cell organelles that are involved, especially the chloroplasts, the peroxisomes, and the mitochondria. They always evidently support something called as photorespiration. Okay. So the rate of respiration actually depends on the increase of light intensity, and it also decreases with the oxygen concentration, and then raised whenever there is high oxygen concentration. Again, it is raised. Now, one second. Yeah. Coming to another cycle, which is called as the Hatch-Slack pathway or the C4 pathway. What is happening in C4 pathway? In C4 pathway, we have a very unique kind of a setup within the plants. So the C4 plants are very efficient plants. They grow in tropical regions. Example, sugarcane. So here they are very rapidly photosynthesizing plants. So what do they do is they the first compound that they fix as a carbon dioxide is a 4 carbon containing compound such as aspartic acid or malic acid. So Hatch and Slack are the ones who discovered this pathway, like that of a phosphoglyceric acid. Here also there were 4 carbon containing compounds such as aspartic acid or malic acid. Here the carbon dioxide fixation happens. Now, to understand the C4 pathway, you have to understand a very important anatomy of the plant, which we call it as Kranz anatomy. So they represent this Kranz anatomy in some of the families such as grasses, sugarcane, maize, sorghum, and tropical grasses, etc. So here, what they do is, these species, I told you that they have a Kranz anatomy. They have vascular bundles. Around the vascular bundles, you have a ring-like arrangement where you have bundle sheath cells which are very, very tightly arranged, and these bundle sheath cells have very thick walls. They're impermeable to gases, and they don't have intercellular spaces, and they have very dense cytoplasm. Whereas around the bundle sheath cells is where the mesophyll cells are there. So the characteristics of the cells are very, very important. Large in size, large grana, okay, and they have agranular chloroplast, and they contain starch grains, very less intercellular space. And you know the mesophyll cells already. They are similar in size, they contain grana, and they lack starch grains. They have bundle sheath cells around them, okay. Now, there is a proper understanding between these bundle sheath cells and mesophyll cells. It's very, very easy to understand. Now, what is happening? Atmospheric carbon dioxide is absorbed through which cells? So here, see, here what do we have? Lower side, we have bundle sheath cells, and in the mesophyll cell on the upper side. Atmospheric oxygen enters the mesophyll cells. Here it is converted in the form of bicarbonate. Now, this bicarbonate mixes with phosphoenol pyruvate. This phosphoenol pyruvate, okay, is used for the fixation. So the HCO3, which is a bicarbonate, combines with the phosphoenol pyruvate, which is a C3 acid, which is a C3 compound. When the fixation happens, it is converted into a C4 acid. Phosphoenol pyruvate gets converted into malic acid or aspartic acid, which are 4 carbon containing compounds. Now, these 4 carbon containing compounds are transported to the next cell, that is the bundle sheath cells. In bundle sheath cells, what happens? You have 4 carbon atoms, isn't it? Among this 4 carbon atom, one of the carbon atom is removed. When this carbon atom is removed, you get carbon dioxide as the byproduct. So these 3 carbon atom containing compound, they form the C3 acid. Okay. So this C3 acid, you know that it is a pyruvic acid. Okay, and the carbon dioxide that is released here, it is used for fixation, like how we saw in the C3 cycle. The C3 cycle continues here again, okay, where in the bundle sheath cell. Now what happens to the C3 acid? The C3 acid again is transported back to your mesophyll cell. The regeneration happen. C3 acid is converted into phosphoenol pyruvate. Phosphoenol pyruvate receives the oxygen, carbon dioxide from the atmosphere, and it fixes the carbon dioxide into a 4 carbon containing compound or 4 carbon containing acid, which we call it as aspartic acid or malic acid, and the cycle continues. So this is called as C4 cycle. Okay, so this is very very important. Why? Because here they do not depend upon the atmospheric carbon dioxide concentration. They just can regenerate their own carbon dioxide. That is why they have a very high rates of photosynthesis. Whereas if you compare the same thing with the rest of the plants, like the bell pepper or the tomato, so this bell pepper or the tomato, they lack Kranz anatomy. Since they lack Kranz anatomy, now what happens, students? You have to put them in greenhouse, greenhouse, where the carbon dioxide concentration is increased, and then they can synthesize without reducing their photosynthetic output. So this is about light reaction and the significance of C4. Now the last part is left, that is the factors affecting the photosynthesis. And factors affecting the photosynthesis we'll discuss. And let me see if you guys have any doubts or if there is any response. All right. I hope it is audible, students. If you have any doubts, please make sure that you post your doubts without any hesitation. We can talk about it. Sometimes when revision classes, the topics are rushed, but it can be. Okay. So let's talk about the last phase. That is, what are the factors that affect the rate of photosynthesis? Now, the factors affecting the photosynthesis are, that it is affected by the light, the carbon dioxide concentration, okay, which are the two important limiting factors. Okay. Now, coming to the light. Now, what happens? Photosynthesis is very dependent upon the light and the quality of the light that is incidenting on the plants. So basically, the shape of the leaves and the leaves that are the way they're exposed to the sunlight, it is very, very important. So during the solarization or whenever the light is present, something can happen called as photooxidation. So for a few hours. So photosynthetic apparatus, they get destroyed. That is why we have for accessory pigments which reduces this photooxidation and protect our chlorophyll. Okay. So during morning hours, we have very mild light and it is very, very helpful for maximizing the photosynthetic output. At very intense light, what happens is usually the gases exchange is also reduced because of excess heat. The stomata gets closed. When the stomata gets closed, the gases exchange reduces, carbon dioxide exchange reduces. When the carbon dioxide exchange reduces, now obviously the photosynthesis is also reduced. Correct, right? So that is why the exact exposure of the light is very very important. It's not all in the same plants, especially in some of the plants which are dark plants which love shady areas. They don't specifically depend on the light. So anyways, I don't want to rush this topic because it's very very important in some part of it. So we'll stop here and we will continue in the next class, okay, with regarding to the rest of the factors that affect the photosynthesis. And then we'll solve some questions and we go to the next chapter, that is respiration in plants. All right, students. So now you can ask your doubts. I will stay here, okay? All right, students, let's close the session then. If you don't have any doubts, we'll continue the topic tomorrow.